Hi, today we're going to talk about DNA, and we're really just going to do a fast overview of the structure of DNA and DNA replication, because you guys have had information about DNA in biology, you've had it in 7th and 8th grade, so on and so forth. So we're just going to do kind of a quick overview just to make sure we remember everything we're supposed to. So some of the uses of DNA, and we're talking about things that we use DNA for in the outside world, is it identifies suspects and victims. So we're able to use it to identify suspects and victims in like things like the World Trade Center situation, bombings, anything like that. You can use DNA. It's able to clear the innocent. So it's there's a project called the Innocent Project, and they go back and they look at cold cases and they run DNA from people who are put in prison who would say that they were innocent. And now that they've developed DNA profiling to such an advanced level, they go back and they take a look at some of these cases, and it allows them to clear people who were innocent of a crime that might have been wrongfully convicted. It's used to establish paternity. So it's using paternity tests to figure out if the father is actually the father or if it's somebody else. And it's used to match organ donors. So if you were somebody who needed a kidney transplant or something like that, they would be able to match organ donors with your DNA. And the location of DNA in the body, right? It can be extracted from any of the following: blood, from your white blood cells, semen, urine, bone, hair follicles, saliva. Okay, so lots of places in the body that you could get or pull DNA from. So the main goal of DNA, the reason why we have it, is to store genetic information that will eventually be translated into a protein, right? You go through the process of RNA, and then you go through tRNA, and you make a protein that actually does something productive in your body. Your DNA is what stores the information to make the protein. So it's like kind of the blueprint of how to do something, or the directions or the recipe for how to do something that then gets used to make something later on. So just a quick recap. You have 46 chromosomes in your body, 23 pairs, right? You get half of them from your mom and half of them from your dad. Chromosomes are condensed DNA, like right before the cell is going to split into two. Looks like an X shape. A gene is a small segment of DNA that codes for a specific protein. So hopefully you remember that as well. So it's like segments of DNA that code for a specific protein, like the protein for eye color or blood type or hair color or whatever, right? And then the proteins are made from the directions in the DNA, and they actually give you the traits. Like you have a protein that makes your hair red, or a protein that gives you A antigens on the outsides of your red blood cells, or different things like that. So it goes DNA, gets transcribed into RNA, then gets translated into a protein. Okay, so that's kind of the process. And hopefully that stuff is coming back to you. I know you had this all in biology class. So that's why we're kind of just going through it quick, but it's some buzzwords that when we start talking about DNA at a forensic level, you'll want to know what it is. So what is a nucleotide? A nucleotide is the building block of DNA, okay? It's like the smallest part that we have of DNA. So there's 3 parts to a nucleotide: the nitrogen base, the sugar, which is a 5-carbon sugar, and then a phosphate group. And it looks like this: a phosphate group, the sugar, and then the nitrogen-containing base. And you guys probably remember that there are 4 bases, A, T, C, and G. The nitrogen-containing bases are right here. So the phosphate and the sugar is the backbone of DNA, and then the nitrogen-containing base is what changes. And the order of those bases is what makes your DNA unique compared to somebody else's. So again, here's your nucleotides, right? There's 4 different nitrogen bases. So every nucleotide has a phosphate, a sugar, and then one of the bases. You can have a purine, which is adenine and guanine, your A or G, and they're 2-ringed. Or you can have a pyrimidine, which is thymine and cytosine, and they're single-ringed. So you would have like a phosphate, a sugar, and then adenine attached right here. Or a phosphate, a sugar, or guanine, so on and so forth. And you'll remember that some of these pair with each other, right? A's always go with T's and C's always go with G's. So you always have a double ring with a single ring because it's just kind of how the bonding works. So Chargaff's rules, we kind of just talked about this, but in DNA, Chargaff was the guy who discovered that all DNA contains the same amounts of adenosine as thymine and the same amounts of cytosine as guanine. So there's always the same amount of A's as T's and C's as G's. So what that means is A always bonds with T and C always bonds with G. So if you have an A, there's going to be a T, there's a T, there's going to be an A, so on and so forth. So DNA is like a twisted ladder. Its shape is called a double helix. That'll be important when we start talking about replication and when we start talking about the polymerase chain reaction. And so it looks like, it kind of looks like this. I'll show you a picture of the double helix in just a second. But here's kind of how it works. The phosphates and the sugars are the outside, and then the rungs, or the inside of the ladder, are the nitrogen bases. So you have A's and T's together, and you have C's and G's together. And so you've got an outside of phosphate sugars here, an outside of phosphate sugars here, and on the inside you have your bases. A's with T's and C's with G's. It looks like this. So this is the double helix shape right here. Okay. So complementary base pairing. Okay, it just means wherever there's a T, you put an A. Wherever there's an A, you put a T. So right here, go ahead and try this strand. Try making right next to it an opposite side strand. So if this was one side of the DNA, what would the other side look like? So for example, if there was a T here, there should be an A. If there's an A, there should be a T. If there's a C, there should be a G, right? So it should look like this, correct? Okay, that's called complementary base pairing. Wherever there's one letter, the opposite letter goes. So T's always go with A's and C's always go with G's, so you always know which letter to pair up. That's going to be important when we talk about DNA replication. That's it. That was your quick overview. I know it was fast, but it's all stuff that you've learned before, and I just wanted to make sure and kind of go through it and refresh your brain. I also want to take a look here at DNA replication because this was also something that you've learned in biology, but I just want to refresh your brain. So DNA replication is copying the genetic information of a cell. So when your cells divide with mitosis or meiosis, right, you have to copy the DNA so that both cells get a copy of it, and the process of doing that is called DNA replication. So we're going to go through the steps of DNA replication really quick and just refresh your brain. It's going to be important when we get into the process of DNA profiling. That's why I'm going over it with you now. So the first step in DNA replication is to unzip the DNA double helix. So remember we said that the DNA is in a double helix, so twisted ladder shape. Well, we need to pull that ladder apart so that we can get to those bases. And the molecule that does this is called helicase. And so what happens is the helicase molecule kind of butts its way in here and pulls the DNA apart so it's no longer twisted and it's opened up. So that's step one. I highly recommend in your notes that you write what the step is and that you draw a picture example so that you have it. Step two, the nucleotides form hydrogen bonds with the template strand. So nucleotides are going to come in here, and so for example, let's see if I can show you this here. I'm going to type it, I think. If there was an A here, and then a T here, and then a C, and then a G kind of coming in right here, the opposite nucleotides, free-floating nucleotides, would be over here, T, and then there might be an A, and then there might be a G, and then there might be a C, just hanging out over here in the cell. They would come in and they would bind in the right spot. So like if there was an A on my strand, a free-floating T would come in right here, see? If there was, the next one was a T, a free-floating A would come in, and it would bond here. If the next letter was a C, a free-floating G would come in. Okay, and if there was a G, a free-floating C would come in, and it would just keep going. So the nucleotides would come in and form hydrogen bonds with the template strand in the correct sequence. okay, and start to make a new strand. Then something called the DNA polymerase, which is a big protein, comes through and it bonds the nucleotides together to form a new double helix. And this step kind of happens simultaneously, like as the base pairs go through and pair up, the DNA polymerase is right behind it and kind of bonds everything together. So you can see like right here, this nucleotide's coming in and the DNA polymerase is right behind it, and it's already starting to twist it back up with the old strand and the new strand. So now you can see that the new strand is like a gold color and the old strand is a blue color. Okay, and what we're getting is we're getting two exact copies of the DNA, each copy with an old strand and a new strand attached to it. So step 4 is the nucleotides form hydrogen bonds with the new strand, and you have two molecules of DNA that are exactly the same. They have exactly the same sequence. Each one has part of the old strand and then the new strand that was attached to it, and then part of the old strand and a new strand that was attached to it. And that's the process of replicating DNA. Now we have two copies of it. If we needed to make more copies, we would do the same thing again. We would make more copies by splitting it with the helicase, adding the nucleotides, the DNA polymerase would bond it together, and now we would have more strands. So here's the steps, right? Enzymes unzip the double helix. Free-floating nucleotides form hydrogen bonds with the template strand by base pairing correctly. The DNA polymerase enzymes bond the nucleotides together to form a double helix, and then they form covalent bonds between the nucleotides to form the new strand. Okay, so here, let me see. Here's what it looks like. Hang on just a second. Let me see if we can pull up. And show you what it looks like.
DNA is a molecule made up of two strands, each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other.
So hopefully that gave you an idea, a visual idea of what it looks like when the DNA polymerase is running through and attaching the base pairs accordingly.